Hi, everyone. It's Michael Yeaman from Los Angeles. I'll be talking about restoring immune tolerance in NMOSD and MOGAD. I'll focus mainly on NMOSD, but obviously the applications are translatable to MOGAD and really any autoimmune disease. Here are my transparency disclosures. I am an advisor for the Guthy-Jackson Charitable Foundation, and I have advised for companies that develop therapeutics for NMOSD. I really want to focus on four discussion points today. First, a brief summary of what might be called the NMOSD revolution, which really is a true rare disease success story. That said, there are remaining unmet needs in NMOSD and related disorders, and those present increased disease burdens that have yet to be addressed. We will also focus on immune tolerance and tolerization. I'll really emphasize this as a brief sort of refresher course, so not extensive detail, but I hope it's useful as a summary. And finally, translate some of this interesting work to the extraordinary opportunity that exists now to restore immune tolerance and potentially arrest, reverse, and possibly even cure autoimmune diseases. So first, a few comments about the success story, which really is a tribute to many of the individuals in the audience and many more worldwide that have been the champions and the pioneers and the engine that has made this story a success. If you were to look at sort of the NMOSD field or the field of NMO and related disorders 15 or so years ago, there would have been some important papers, for example, the discovery of the anti-aquaporin 4 antibody, but there weren't many publications, maybe 100, 150. Fast forward to this year, and now there are over 9,500 publications on NMOSD, with a total citation number of over 200,000 and an H-index collectively in this NMOSD field of 174. That is a remarkable impact on a field. With respect to what, for example, the Guthy-Jackson Charitable Foundation has efforted to do, it was founded in 2008. It held its first International Patient Day in 2009. Its Circles biorepository was launched in 2012, 2013. There are many other excellent biorepositories worldwide, including the NMOS repository, the NOMADMOS, MSBase, and many others. The International Clinical Consortium was formed by GJCF in 2013. Clinical trials began roughly 2014. There was the collaborative publication of the IPND criteria in 2015, and now we're working on an updated version of those diagnostic criteria called IPND 2025. All of this effort led to 4 clinical trials that resulted in 3 approved therapies in 2019-2020, a 4th FDA-approved therapy in 2024, and if you consider rituximab approved in some places in the world, 5 approved therapies, when after 100 years with no clinical trials and no approved therapies, there were no real treatments that were specific for NMOSD, and now there are 5. So a remarkable success story. I think the translation of all of that effort to understanding how it has helped benefit patients in terms of saving and improving lives, I've tried to summarize in this sort of a scorecard. And I'll just be brief here, but if you were to look at the mean average global prevalence in 2008 for NMO and like conditions, it was about 1 in 1 million. Today, it's about 7 in 100,000 on average, and this indicates a 70-fold increase in prevalence. Are we happy that there are many more patients diagnosed with NMOSD and related disorders? No. But we are happy that those who have these conditions are diagnosed much more quickly and much more accurately than just 15 years ago, when approximately 75% of patients had either a delayed diagnosis or a misdiagnosis. Today, that number is more like 15%. So there's been a very nice improvement in diagnosis. Interestingly and importantly, there has been a 10-fold reduction in annual relapse rate overall, based on estimates in the literature. That is really a tribute to the people in the audience and those in the ICC and elsewhere who have really made great efforts to apply these advances in science and medicine to patients and reduce relapse rates. Of course, the pharmaceutical companies are also to thank for developing drugs that reduce risks of relapses. So again, 15 years ago, there were 0 clinical trials. By 2015, there were 4 that had begun. Today, there are more than 35 clinical trials, either active or preparing to launch. And again, now we have 5 approved therapies in the world when just 15 years ago, there were 0. So an incredible success story, and we want to really thank all of those who've been collaborative and collegial and putting all of the effort to making these kinds of advances happen. Other recent highlights in research are also very promising. We know that the FDA-approved therapies reduce relapse risks by 90% or more, and perhaps even increased efficacy over time. We do know that there is a need for full immunization to mitigate serious infection risks that are attributable to some types of therapeutics. There are non-cell-based assays in development and in evaluation to detect anti-aquaporin 4 antibody and anti-MOG antibody and other antibodies, and those are providing some promising results. We are also recognizing the limitations of EDSS-based disability, and some of those metrics are not recognizing disability in NMOSD the way that we need to detect and address. We know that there are some new insights into HLA and other genetic basis for risk or cause of NMOSD and related conditions. More to come on that. There's been a new biomarker, multi-protein biomarker signature identified that appears to forecast relapses months in advance with a very high accuracy. Those efforts are in the works in terms of publication, and we'll see if that holds true. And then very recently, thymic B cells and T cell exhaustion have been implicated in roles for tolerance to aquaporin 4 and also loss of tolerance. So a lot going on, a lot of important research, but just to highlight some of the advances in the success story that continues. So what then are the unmet needs? They're against the setting, the backdrop of the great successes, there are unmet needs that still need to be addressed. So these include the fact that even the best treatments do not prevent all relapses. Among non-approved therapeutics, about 25% on average relapse rate. For the FDA-approved therapies, roughly 13% of patients relapse, maybe as high as 15% on average. And for the overall therapeutics collectively, about 20% of patients relapse. So as good as the latest high efficacy therapies are, still room for improvement. We also know that certain and all NMOSD therapeutics come with risks that include infection and cancer. So urinary tract infections and respiratory tract infections are not uncommon in patients being treated with therapies for NMOSD, including multi-drug resistant, extensively drug resistant pathogens and intero-invasive pathogens, which can cause ascending urinary tract infections and potential sepsis. As far as respiratory tract pathogens are concerned, infections include pathogens that are multi-drug and extensively drug resistant, and oftentimes capsule positive pathogens cause sinopulmonary infections, which can be recurrent and difficult to treat. There are some life-threatening infections that we have to be aware of, including Neisseria meningitis, or meningococcal disease. These can be increasing in their resistance to multiple drugs, and some of them evade vaccines. So we have to be really vigilant about using the best practices to mitigate infections caused by these pathogens. Of course, latent viral, viroid, or even prion infections are known to exist and perhaps can be unmasked in the setting of therapeutics that suppress the immune system. Likewise, cancers, including lymphomas and other cancers, may occur or be at increased risk in patients treated with NMOSD therapeutics, as these cancers have proliferative phenotypes that would otherwise be checked by immune surveillance, but in the setting of immune suppression, that goes unchecked. So there are some risks that we have to address, and again, room for improvement. There are many active clinical trials for evaluation of NMOSD candidates going on worldwide. That's good news. And at the same time, many of these will have the same risks and challenges as the approved therapeutics do today. So collectively, that brings us to the idea of what can we do to go beyond treatments that suppress the immune system generally. And this brings into focus the idea of immune tolerance. So a quick refresher course, what is immune tolerance? Well, it is in essence how your immune system protects you against internal or external threats such as cancer or infection without harming any healthy molecules, cells, or tissues that make up the body or that allow good function of the body. Another way to say this is that immune tolerance is the system of checks and balances that allows the immune system to do good, but prevents the immune system from doing harm. So we definitely want the immune system to do some good things. For example, we want it to make sure that the molecules and cells that we have in our bodies are healthy and normal. We want the tissues that express and display these molecules to be healthy. We want wound healing to be optimal. We don't want it to go off track and cause problems. And we want normal growth and development. We also want the immune system to reject pathogenic microbes, to detect and destroy precancerous cells and cells that are likely to lead to cancers or tumors. We want the immune system to mediate proper wound healing and reject poor wound healing, for example, fibrosis or abnormal types of wound healing. And we want the immune system to keep on track the detection of abnormal growth that might be adverse to health. Well, one of the most important cells and cell subsets in immune tolerance is the T cell. And one of the places where the T cell has its greatest influence for tolerance is in the thymus. So I like to think of the thymus as the T cell university. And it's because when the T cells and other lymphocytes leave the bone marrow, T cells traffic to the thymus. And this is an organ that's about the size of your fist. It's folded around the esophagus just above the heart. And it is composed of several different contexts. First, the outer layer, the capsule, then the next layer inward is the cortex, and finally the medulla is the deep internal part of the thymus. T cells undergo 3 examinations, if you will, in each of these places in the thymus. So in order for T cells and B cells to be allowed to graduate from these universities, the case T cell university for the thymus, they have to really pass these 3 tests. First, can they recognize antigen-presenting cells? This involves a molecular handshake between presentation and reception between a T cell and an antigen-presenting cell. It might involve things like primary signal or second signal ligand receptor interactions. If the T cells can do this properly, they advance to the next step. If they cannot, they are deleted or censored. The next test is, can the T cells have a proper molecular handshake with the antigen presenting cell? This involves T cell receptor, MHC, epitope recognition, along with checkpoint inhibitor interactions that are appropriate. If that happens, these T cells move forward. If not, they are censored or deleted. And finally, the last exam is, can the T cells respond to an antigen in a way that is optimal, not minimal or maximal? And this involves more nuanced interactions, including the degree of binding, for example, of a T cell receptor to an epitope, a peptide sequence from an antigen. Too strong of binding or too long of binding, that is likely associated with an autoreactive T cell. And so those are deleted if there's inappropriate interaction. But if they pass muster, they are allowed to graduate. Please keep in mind that only about 1% of T cells successfully pass all three exams. So it's a pretty stringent university system for T cells to graduate. But those that do traffic to the lymphatics. And in particular, T cells leave the thymus and find their way to lymph nodes, where they reside and wait for antigen-presenting cells to come to them often and present the antigens that they have found in the periphery. And at that point, the T cells are either activated or energized in such a way that the active responsive T cells leave the lymph node and move to tissues where they might be most effective in terms of conditioning or governing immune paradigms for response. Well, with that as a backdrop, how is immune tolerance lost? The answer is that we don't exactly know. But many of the steps involved in tolerance that I just mentioned probably come into play in terms of potential mistaken identity, where antigen-presenting cells might misrecognize an antigen, or the interaction between APCs and T cells might be dysfunctional, or T cell B cell interactions, or a combination of these problems. It's also very possible that the antigen itself can be altered in a way that triggers or is mistaken as a potential threat. So all of these are possibilities. Just briefly, let's review sort of the process by which this might occur. So we know that astrocytes express aquaporin 4, for example, in the case of NMO. Aquaporin 4 is detected and processed by antigen-presenting cells like dendritic cells and others that will display pieces of aquaporin 4 on their surface in the MHC context. Well, these antigen-presenting cells present the antigen to a naive T cell, but they do so in context of two interactions. One is this molecular handshake between the epitope and the T cell receptor, but also other ligands that are secondary signals and checkpoint inhibitors. And this is all done under the conditions of certain cytokines. For example, if this interaction is conditioned by IL-6 and TGF-beta, this T cell will polarize to a Th17 response, which releases IL-17 and other cytokines. Now, activated T cells then do a next wave of functions, which include a couple of different pathways. First, for example, Th17 T cells will release IL-17, which upregulate neutrophil maturation and activation. I'll come back to that in just a moment. But these T cells also interact with B cells in a way that is bidirectional. The B cells influence T cells, for example, by way of IL-6, and T cells can also polarize to a type 2 function, which is mediated largely by IL-4 or IL-13, such that promotes CD20 to CD19 B cell maturation, upregulation of antigen-specific antibodies, for example, anti-aquaporin 4 antibody, which moves through the blood-brain barrier, and engages aquaporin-4 on astrocytes. This triggers classical or antibody-mediated complement fixation that results in the membrane attack complex harming or injuring the cell directly, but also the generation of chemotactic factors like C3a and C5a, which move into the periphery and activate neutrophils to marginate and diapedese, such that they, along with other granulocytes like eosinophils, contribute to the inflammatory milieu, which kills or injures astrocytes and results in demyelination. So these are the steps at which immune tolerance can be lost, any of these steps. You could imagine then a logical follow-on would be that you could get loss of immune tolerance due to abnormal antigen structure or expression, for example, ectopic expression. You can imagine that there would be dysfunctional antigen presentation. For example, the HLA or MHC class that does the presenting of antigens from antigen-presenting cells to T cells. Dysfunctional T or B cell receptor genes that engage presented antigens from an antigen-presenting cell. It's also very probable that infectious disease pathogens, neoplasia, or cancer can create molecular mimics that lead to mistaken identity of otherwise healthy proteins or cells. And things like pregnancy or allograft transplantation can result in neoantigens that reverse antigenic ignorance or otherwise might contribute to autoreactive responses. So in that background, what might we do to restore immune tolerance? And that's really where the field is heading in terms of going beyond treatments. So basic strategies to restore immune tolerance, including correcting gene mutations or dysfunctions. And in the advent of the CRISPR technology, this is becoming more and more feasible. Also correcting the production of protein structure that might otherwise be abnormal, including folding of a protein. So a three-dimensional epitope, not just a linear epitope. We also know that potentially reprioritizing how antigen-presenting cells, T cells, or B cells respond to antigens, or a process that might be called immune deviation, is a strategy for restoring immune tolerance. Of course, deleting autoreactive antigen-presenting cells, T cells, or B cells via censoring or deletion might be a strategy. And we know that that's already being done by way of CAR T cells. I'll come back to that in a moment. But what we're really excited about is the potential to re-educate the immune system so that antigen-presenting cells, T cells, and B cells that in the autoimmune state consider an antigen to be foreign, even if it's self, might be retrained such that it no longer sees a self-antigen as being foreign. And that really is the process of tolerization. There are many technologies now that are emerging in the tolerization space. I'll just go through a few of them in a very high-level summary. So we know that dendritic cell vaccines are being explored, and I'll mention an example of this in a moment. Antigen-specific toleragens are decoy antigens or antigens that look like a particular autoreactive antigen but cause an anti-inflammatory response. Some of these are called emotopes or apoptopes. There are also antigen-specific tolerizing nanoparticles, where the autoantigen target is placed inside of a nanoparticle, but the nanoparticle conditions the immune cells to treat this as a safe antigen and induce a tolerogenic response rather than an inflammatory response. Of course, CAR T and AA CAR T cell therapies are now being used with some promise to delete T cells and B cells that have a particular marker on them, and I'll mention an example of that in just a moment. Normally, checkpoint gating is important to prevent an overly aggressive immune response. So, for example, in cancer, checkpoint inhibition is used to allow the immune system to chase down cancer cells when it wouldn't otherwise. But in the case of tolerization, we would be doing checkpoint induction or checkpoint gating, where we actually turn up checkpoint function rather than turning it down like is used in cancer. And by the way, checkpoint inhibition in cancer therapy can cause autoimmune disease. So we really want to be careful and focus on checkpoint induction, but antigen-specific checkpoint induction. There are interesting new approaches that involve leveraging how the immune system tolerates fetal tissue during pregnancy. And this uses the HLA-G mechanism. Inverse vaccines. So instead of classical vaccines that turn up the immune response to a very specific antigen, it turns down the immune response to that specific antigen. We know that there's been some success in bone marrow rebooting, bone marrow transplant, et cetera. The early successes may not be as effective in the long term as had been thought. More to follow, but that's more of an extreme approach where you have to condition the immune system in ways that can cause risks, at least in the acute phase of therapy. There are other types of endogenous tolerogenesis, including the potential for using targeted liver macrophage tolerance, Kupffer cell tolerance, or even red blood cell tolerance mechanisms that might be harnessed to reduce or reverse autoreactivity to specific antigens causing autoimmune disease. And finally, there are some interesting genetic approaches that use DNA to reprogram autoreactive cells through the process of programmed cell death. So lots going on in the field, a lot more to be aware of as we move forward. But just a quick couple of examples of how some of these work. So CAR T or AA CAR T cell therapy is not truly tolerization in the sense that we might think of it, but it can lead to tolerization in ways that we don't fully understand. Here, healthy T cells from patients are removed. They're engineered in the laboratory by way of DNA, chimeric antigen receptor integration, such that these cells will now express receptors for a very specific antigenic target, like CD20, for instance. And then when returned to the patient, they will chase down and kill cells that have that specific antigenic determinant on them. So this is a process of deletion or censoring of autoreactive T or B cells. It's been used, you've probably seen, in lupus, in pemphigus, and in even in NMO in the last few years. By comparison, tolerogenic nanoparticles involve nanoparticles that contain pieces of autoreactive antigens. They are embedded in a nanoparticle that has a tolerizing feature to it. I won't get into the chemistry of that, but when dendritic cells process and display antigens presented in this manner, these are tolerogenic dendritic cells that then go back into the immune repertoire and tolerize B cells and T cells to that specific antigen. And lastly, inverse RNA or DNA vaccines, very interesting. A lot of the technology has derived from the COVID vaccine technologies and those technologies even prior to COVID-19 vaccines. But instead of vaccines that upregulate an immune response to prevent an infection, inverse vaccines introduce nucleic acid that encodes the autoantigen. It can be either DNA or RNA. But this particle, or the strategic modification of the nucleic acid, turns this particle into a tolerizing DNA or mRNA nanoparticle. For example, mRNA that has been modified using pseudo-uridine type approaches can lead to dendritic cells and other cells that react to this vaccine, this inverse vaccine, so as to become tolerizing. And in so doing, actually then go on to tolerize T cells and B cells to that specific antigen. So very interesting, very exciting advances and just emerging. I just want to say a few last words about clinical trials. A lot of work going on right now in clinical trials, early stages for the most part, but we know that bone marrow transplant has been successful, at least to some extent in the past. Whether that turns out to hold true, still in question. There are some interesting new approaches, for example, delayed tolerance, where in addition to a graft implant, the patient is provided with a very specific subset of the donor peripheral lymphocytes in such a way that is believed to introduce a type of tolerization that can help avoid graft rejection. We know that there are early clinical and preclinical advances in tolerogenic nanoparticles that show promise in autoimmune diseases, including celiac disease and others. Many of the dendritic cell-based vaccines that have been tested so far in the clinic have shown safety and good tolerability by patients, and they do induce biomarkers of tolerogenesis, but those are still in the early stages. There's sort of a recent surge in the interest of daratumumab, anti-CD38 mediated tolerization, but that remains to be fully explored. And as I mentioned, some very promising early results with CAR-T therapy in pemphigus, lupus, NMO, and other conditions of autoimmune mechanism. We're really interested in focusing on those going forward, but CAR-T is really not a purely tolerogenic mechanism so much as it is a cell deletion mechanism. But there's a lot we still need to learn. In these regards, NMO really presents an ideal model for immune tolerization, as does MOGAD. In NMO, we know the autoantigen, aquaporin 4, we know many of its critical epitopes. The pathogenic mechanisms and pathways are becoming more fully established. There is a defined clinical phenotype, especially for aquaporin 4 positive NMOSD, and we're working more and more closely to create a standardized relapse adjudication so that we can better design clinical trials. There's a robust and increasing prevalence of this disease, as you know from my earlier comments. It does maintain an orphan disease status and fast track opportunity for novel technologies. And there's increasing interest that we've been cultivating in the biotech and venture spaces. So a lot of exciting stuff going on in this field. I do want to just mention briefly that there is a potential model for tolerization trial design that emerged from one of the consensus meetings at the most recent Guthy-Jackson Summit for NMO tolerization. And it involves a very dynamic process that prevents any patient from being on pure placebo. So here, patients would be enrolled based on inclusion and exclusion criteria with clearly defined endpoints. They'd be randomized into one of two general groups, either standard of care alone or standard of care plus the experimental regimen. Only when biomarkers of tolerogenesis emerged in the experimental arm would the standard of care begin being tapered, so that by the later stages of the trial, the patient would be on experimental regimen without standard of care. And as you can see, at no point would the patient be on pure placebo control. So these are just examples of some of the thinking that's going into clinical trial design for immune tolerance studies. I just want to emphasize as a last point here that compared to immune suppression, which basically asks the immune system to cease its functions or stand on the sidelines, tolerization asks an immune system to actively participate in a new process of peace. And that's really, I think, what we need in the field in a way that goes beyond treatments toward cures. And that's what our focus is, as well as many in the field. Again, I want to thank everybody who has contributed to the great advances in the field of NMOSD and who are participating now in the exciting new era of tolerogenesis. thank you very much for the invitation. I hope this has been helpful. If you have any thoughts or discussion points you'd like to share, please feel free to email me. And I look forward to seeing everyone soon. Thanks again, and hope all is well there.